And welcome to day 2 of the Connect Science webinar. It's a tremendous pleasure to welcome Matthew Meyerson to speak to us on does the microbiome promote human cancer. Dr. Meyerson is an investigator in the Department of Medical Oncology at Dana Farber. He directs the Center for Cancer Genomics at Dana Farber with Bruce Johnson. He's an institute member in the cancer program at Broad Institute and a professor of genetics and medicine at Harvard Medical School. Dr. Meyerson has made seminal discoveries related to the genetics of cancer that extend at least as far back as the identification of EGFR mutations in lung cancer with Dr. Bill Sellers, and it's a tremendous pleasure to have him here. I'd like to make a couple of announcements before we start. First of all, thank you everyone for coming. We had well over 1,600 attendees yesterday, and I expect we'll exceed that today. We have a remarkable list of speakers over the next couple of weeks who've already scheduled their talk, so please do visit the website you see on the opening screen to look at that list of speakers. Tomorrow, we do not have a lecture. We're going to skip Wednesdays going forward because of internal meetings with Dana Farber. But on Thursday, we're very fortunate to have Dr. Stu Orkin, who will be discussing his seminal work related to the activation of transcription factor and its effect on sickle cell anemia, which has been realized clinically by millions of people around the world already. The talks are being uploaded to the website, and you can download Dr. Kalin's talk from yesterday on the website. Dr. Meyerson's talk will be made available later today. And please do feel free to post your questions through the Q&A feature on the bottom of your screen. So I'd like to thank Dr. Meyerson again for joining, and Matthew, please take it away.
All right, well, David, thank you very much for this kind introduction. I just want to make sure that I can both be seen and heard. You should see a slide that's a title, Science Logo. And then you should also potentially see a thumbnail of me. I'm social distancing here, sitting in my kitchen. So real pleasure to have all of you here, and you're joining and paying attention. And I really want to give special thanks to David Weinstock and Nellie Polyak for conceiving of and organizing this seminar series, and to Richard Oakley and Joellen Fredericks at Dana Farber for organizing it and making this connection happen. Okay, so my title here is Does the Microbiome Promote Human Cancer? Contrast to the presentation from Bill Kalin yesterday. This is really, I would say, work of uncertain significance, right? I think it's hard to say if a lot of what I'm going to be presenting today is of great importance or is just an epiphenomenon. And hopefully, work over the next few years should enable us to answer this question. Just first want to mention financial conflicts of interest and of particular significance. We have patents and patents pending on computational subtraction and Fusobacterium, which are not licensed. So the outline of the presentation, going to talk about background on viruses and bacteria and cancer causation, approaches to pathogen discovery in cancer, the colorectal cancer microbiome and Fusobacterium, some studies in other cancers, including lung cancer and esophageal squamous cell cancer. So we may take out the BPDCN work, collaboration with Andy Lane for reasons of length, the optimistic consortium on the colorectal cancer microbiome, together with Wendy Garrett, and some work from Hans Klaver's lab on PKS positive E. coli, and finally a brief comments on summary and future directions. If you hear barking in the background, those are our dogs. I think I'll consider it an expression of enthusiasm. All right, so just a background on viruses and bacteria in cancer causation. There is the first real example of a cancer virus was actually found in chickens, going back to the year 1909. A chicken farmer from Long Island brought in a hen with a tumor on her neck to the laboratory of Peyton Rouse of Rockefeller University. This is a photograph of Dr. Rouse at his microscope, and this is the index case with a tumor. And in this study, he showed that there's a transmissible sarcoma that he could inject from chicken to chicken. And in fact, he showed that critically, it was transmissible by a cell-free filtrate, which was the first evidence that it was a subcellular particle or a virus. And the Rouse sarcoma virus, as you may know, has had a really important history in cancer research. Howard Temin and Harry Rubin showed that this virus could transform 47 years later, a long lag time, could transform chicken fibroblast cells. It required the development of mammalian cell culture to be able to do this. Howard Temin went on to actually use this same virus to identify a reverse transcriptase, along with David Baltimore. Studies of the Rouse sarcoma virus led to the identification of its oncogene, VSARC, and then to the cellular proto-oncogene CSARC by Bishop and Varmus and colleagues, including Dominique Stella, which was the impetus for the discovery of multiple cellular oncogenes and, in fact, the somatic mutation of oncogenes. So really important history in cancer research. There are a number of major cancer viruses that have been discovered. The hepatitis B virus was discovered in 1965 by Barry Bloomberg of the University of Pennsylvania using a serological approach. He found an antibody that recognized a surface antigen of the virus, first called the Australian antigen, which led to the identification of the hepatitis B virus and eventually to vaccination. And that vaccine has led to significant decrease in the rates of liver cancer. This is an example from Taiwan, where in each of these cohorts of ages, and these are just in children and young adults, the impact to reach all age cohorts, you can see a significant decrease in the vaccinated in black compared to the unvaccinated in clear cohorts in each of these age groups up to the mid-20s, with the arrival of hepatitis B vaccination. Similarly, human papillomavirus vaccination is also beginning to have a big impact. Human papillomavirus was discovered in cervical cancer acid hybridization methods by Harold Zerhausen and colleagues working in Heidelberg. In 2006, following the really elegant work of Lowy and Gentler, working at the United States National Cancer Institute, human papillomavirus was introduced into use in the United States. And this vaccine is beginning to lead to significant decreases in cervical cancer incidence in young adults, in young women in the United States, shown here after this join point here in 2009. You can see a rapid decrease in rates of cervical cancer, and we can anticipate these will continue and as the vaccination takes hold and becomes more widespread. Finally, in major cancer viruses, I want to mention the Epstein-Barr virus, really found through epidemiological approaches. Dennis Burkett, working in Uganda, found rapidly growing lymphomas, first of the jaw and then other locations and it's turned out was throughout Central Africa, leading to the identification of viral particles by Anthony Epstein in the UK. The Epstein-Barr virus is associated with lymphomas and nasopharyngeal carcinomas. Recently, as part of the work of Genome Atlas, led by Adam Bass, Epstein-Barr virus positive gastric carcinomas associated with immune activation and a risk to immune checkpoint inhibitors were shown to be one of the 4 major types of stomach cancer. In fact, found to be present in about 10% of stomach cancers. It has yet been approved Epstein-Barr virus, so I think that's a really important topic. Turning to bacteria in human cancer, this was work of Robin Warren, a pathologist, and Barry Marshall, a gastroenterologist working at the University of Western Australia in Perth. They started by identifying a novel bacterium in the stomachs of patients with gastritis. Patients with Helicobacter pylori have about a 6 to 8-fold increase of non-cardiac gastric cancer compared to patients with H. pylori. And one interesting thing about H. pylori is that it's not enriched in the cancers themselves. And so it's likely that there's going to be a role in initiation rather than progression. And this may apply to other cancer causations as well. Interestingly, Barry Marshall really wanted to prove his hypothesis about Helicobacter pylori. You can see him in this picture raising a glass together with Barry Marshall. He, in fact, drank a flask of Helicobacter pylori, gave himself gastritis, and treated it with antibiotics. I just want to say, I don't think any of us working on the colorectal cancer microbiome are planning to do that same experiment, but it really a lot of boldness. So I'd now like to talk about our approaches to pathogen discovery in cancer. I'm going to talk about some work not shown here is a picture of Alex Kostic becoming late Weber and both students who actually collaborated with us. Chandra Petamalo, a former computational biologist, Akina Ojishina, a former postdoc, and Mark Walker, computational biologist at the Broad, have made comments to our method there with Alex Kostic, whose picture will a bit later when I talk about Fusobacterium. So I mentioned a number of discoveries over time, and different technical advances have led to the discovery of chronic disease microbes. So for example, human papillomavirus, HPV 16 and 18, and found by nucleic acid hybridization methods. The key technical issue for Helicobacter pylori was advances in microbial culture, in particular, the development of microaerophilic techniques. And finally, really of Kaposi sarcoma by the use of PCR-based subtraction or representational difference analysis to find this virus associated in HIV-associated disease. So this was a real inspiration to us, as I mentioned. In fact, we tried this PCR-based subtraction method in our lab. Technically very difficult approach, and this was actually an inspiration for the approach that we developed subsequently, which we called seq-based functional subtraction. And the principle in infected tissues contain both human and microbial RNA and DNA, and the microbial sequences can be identified by subtraction of human sequences. So you generate and sequence from human, and then with subtraction, left with these red sequences, which are of non-human origin, and the disease-specific candidate sequences can be validated experimentally. Work led by Griffin Weber, Jay Shanduri, who was a student in George Church's lab, and David Tannenbaum working in my lab. At that time, sequencing was incredibly expensive, but as you know, there's been an extraordinary decrease in sequencing costs, which has really enabled now large-scale computational subtraction and analytic approaches. So in fact, the first real advance using a method similar to ours was the discovery of Merkel cell polyomavirus using digital transcriptome subtraction by the Chang and Moore laboratory now at Pittsburgh, who used high throughput sequencing and computation. They showed in their first paper that the virus is clonally integrated into the cancer genome at different locations, as you can see on the southern blot on the right. And there's really been, since then, a lot of remarkable work on this topic, including a lot of beautiful work by Jim DiCaprio, for Cancer Institute, who've done a lot of work on genomics and functional analysis of Merkel cell cancers, including a lot of functional studies of the role of the Merkel cell polyomavirus. Developed our methods, Alex Kostic, working with Akina Ojishina, really updated our computational methods for deep sequencing of human tissue, taking those computational methods from their previous approach in conventional sequencing to really having the capability for high throughput sequencing. So it required a variety of filtering steps, including quality filtration and what we're continuing to do, removal of low complexity reads, alignment steps, multiple alignment steps, in this case using MAC megablast, and blast. Finally, which gives you an unmapped read set, which then you can use to perform for metagenomic analysis on known organisms or for further analysis on unknown organisms. Since then, Chandra Peddamalu and Mark Walker have really extended this. They have a publicly available software that you can download known as GATK PathSeq. And this is similar, except that the key difference and increasing speed is used by comparison against a host KMER file. So for example, take 4 dimers from the human genome as your subtraction. And so this gives you a very rapid, efficient subtraction method. And this is available to the public through GATK. So Alex did the first week, and then his work in collaboration with Ami Bhatt and others, and the work of Susan Bowman, all former students or fellows in my laboratory, on the identification of Fusobacterium in colorectal cancer and studies of the function of Fusobacterium. Our first study was done in collaboration with Adam Bass, now on the Dana Farber faculty, as well as with Josep Tabernero, major contributions from Mike Lawrence, who was a postdoc in Gadi Getz's lab and is on the Massachusetts General Hospital faculty. This was the first sequences of colorectal cancer normal genome pairs. I should actually just say a little story about this. When Alex completed PathSeq, I said, Alex, this is wonderful, but what do you want to work on next? I said, Alex, I think you should think about diabetes. To it. Why don't you go on that? I said, oh, Matthew, that's too hard. It may not be present when the patients have disease. We can't get samples. I don't think it makes sense to study diabetes. He said, let's not do that. Instead, let's look at colorectal cancer. And I said, oh, Alex, that's too hard. The colon, it's filled with bacteria. You're just going to get background. He said, no, Matthew. The colon filled with bacteria, so maybe one of them could cause it. Oh, all right, I'm persuaded. Why don't you go ahead? And he did this analysis. And I just want to mention that right now, Alex is now on the faculty at the Joslin Diabetes Center looking at microbiome correlates with diabetes. So maybe that station had impact going both directions. So he took 1.5 billion reads from these colorectal cancer genomes and subtracted down to 100 reads. And then method developed by Nicola Segata, who was then a postdoc at Curtis Huttenhauer's lab, now is in Torino, and looked for enrichment in populations of tumors. And initially, we were very excited with Fusobacterium nucleatum and also of Streptococcus. And there was a long-standing association of Streptococcus bovis with colorectal cancer. We actually did not find this association of Streptococcus held up in our further studies, but it was there initially. Statistics of the Fusobacterium, actually, if you look at tumor normal pairs, the normals are in green, the tumors are in purple, and you can see pairwise enrichment, in many cases undetectable in the normal, detectable in the tumor, in some cases detectable in both normal and tumor. This is adjacent normal from the same patient. If you look at uninvolved patients with resections of the colon for other reasons, there's almost no Fusobacterium in the normal colon and not a lot of in the normal stool, and I'll come back to that later. Alex went on to confirm this by ribosomal 16S DNA sequencing of 90 colorectal cancer normal pairs. In fact, again, he saw these are using the same analysis using FC from the Huttenhauer lab. You could see enrichment of Fusobacteria on the species, genus, phylum levels, and all, but not Streptococcus. And then if you look at the whole microbiome, the only group enriched in tumors was the Fusobacteria. I should mention in parallel to our discovery, There was a similar discovery from the laboratory of Robert Holt, working at University of British Columbia and the British Columbia Genome Sequencing Center, first author Diego Castellaran, and we published our back-to-back with the paper from the Holt Laboratory. Alex went on to start in situ analysis, and this experiment was our collaboration with Wendy Garrett's laboratory. And you can see here, you can detect here, you can see very clearly a universal bacterial eubacterial probe seen within a colon cancer. And you can probably see faintly here in red, Fusobacterium-directed probes can also be detected. And I'll show some more data on that in some slides. One of the questions is, when does Fusobacterium first associated with? Alex went on to, by qPCR, to show that actually Fusobacterium is enriched not only in the colorectal carcinoma stage, but also in the adenoma stage. And that's shown here. Slide, we can see here at the CT values, difference between Fusobacterium and bacterium that are overall in the adenoma. So I should say, in fact, the difference in delta CT value is larger in the normal adenoma, representing more amplification in the adenomas. Furthermore, by a similar measurement, you could see an increase of Fusobacterium, again, a delta CT value here in the carcinoma compared to the adenoma compared to the norm in patient stools. So significant increase in Fusobacterium abundance in carcinoma compared to normals, but see so that this alone doesn't appear to be sufficient to be a diagnostic test, although it's possible that for patients with very high levels of Fusobacterium in the stool, this could be potentially valuable as a test. Alex went on to collaborate with Wendy Garrett's laboratory to ask if we feed Fusobacteria to colorectal cancer mice, are we able to promote tumorisis? I think, in my view, the answer is still open to this question. But this experiment provides some evidence that, in fact, you do see promotion of tumor genesis. And this is a colon tumor count in CMIN mouse treated with daily feedings of Fusobacterium nucleatum or of Streptococcus sanguinis or of a sham control. I have to say these are pretty difficult experiments. It's a lot of work, right? Daily oral gavage, Fusobacteria smell terrible anaerobes, constitutive anaerobes. And so, hats off to these experiments. You can see there's a statistically significant increase of the number of colorectal tumors fed with one particular Fusobacterium species, one highly invasive species. We did not see this in a couple of mouse models. In contrast, an IL-10 knockout mouse, T8 and RAG2 knockout mouse, which have high colitis scores, but no increase in colorectal tumors. We thought that we might see an increase in these models, but in fact, we didn't. So I think this provided some evidence that Fusobacterium can promote colorectal cancer, and I think there's increasing evidence that's coming from a variety of groups that's consistent with that result. One of the questions that asked early on was, are Fusobacteria present in colon cancer cell lines or metastases? And so he looked at this with PCR. He found fusobacteria are not present in the cancer cell lines. In contrast, and they're not present in normal livers that are cancer. In contrast, he did detect fusobacterium colorectal cancer metastasis, and in one case, in normal liver adjacent to a metastasis. And I was honestly kind of skeptical about this result because it's a, it's a. Sorry, trying to adjust here. That's the problem. Can you? I just want to make sure I can be seen, and it looked like for some reason my video wasn't on anymore.
Hi, we
So
turned off your video just to help out with the audio distortions we were hearing.
Got it. Okay, you can turn it back off.
Okay, turning it off now.
Okay. All right. Yeah. Sorry, I was just trying to figure out why I couldn't see the video, and I hope this is through with enough bandwidth. So I was a little skeptical of this result because it was a qPCR when Susan Bullman joined the lab. Susan had a background in microbiology, and so here we go. And she was actually able to culture fusobacterium from different specimens, and eventually she developed the ability to culture fusobacterium from flash frozen specimens, which I think was pretty remarkable. When she did this, what she found, and again, I thought this was a pretty remarkable result, that when she cultured bacteria from either primary colon tumors or that Joseph Tabernero and colleagues have built at the Vall d'Hebron Institute of Oncology in Barcelona. So what you can see is that right in these pairs, she could culture both from the colon primary and the liver metastasis, and even where there was material for culture, she was able to detect by qPCR. In the cases that are fusobacterium negative, they were also negative in the liver metastasis. And then she sequenced this. One case was a fusobacterium necrophorum, and the other case was a fusobacterium nucleatum. And you can see that these are very identical if we take percent identity of different genes between the primary and the metastasis. And in fact, Susan went on to do RNA sequencing and DNA sequencing of the liver metastases. And here, again, she looked at relative abundance of bacteria between primary and the liver metastasis, and she found a collection of organisms be present in both. They included fusobacteria, Bacteroides species, Bacteroideus, especially shown in dark, and Vitella species, for example, shown over here in baby blue, and in some cases, Selanemonas butygena, shown here in the purple. So it wasn't just that the fusobacteria were present in the metastasis to the liver, but the whole microbiome from the original sample was also close to there. And in fact, then when Paolo Nucifero, working at Vall d'Hebron in Barcelona, did in situ hybridization for bacterial DNA, you can see here that it's present in or very close to colorectal cancer cells, both in the primary colorectal cancer, as shown here in this left panel, and also in the metastasis to the liver, as shown here in this right-hand panel. Susan went on to study mouse xenografts, and these were xenografts that had been developed by Kimi Ng, Andy Aguirre, Max Loda, Eva Sasinska, Charlie Fuchs, and collaborators at Dana Farber. And she was able to culture fusobacterium, the mouse xenografts of colorectal cancer for multiple generations. Again, this was I thought, a pretty remarkable result. So multiple passages within the mouse, the fusobacteria are still there. And then when she did sequencing of the microbiome, when she did shotgun sequencing and analyzed the microbiome, you could see here that Fusobacterium nucleatum Salmonella, and Bacteroides fragilis are all continuing through multiple generations. Enormous persistence of the microbiome. This suggested to us, look what happens. We can treat these xenografts with nitazole. You can see when treated here, and we've flipped direction of showing the delta CTs for qPCR, but you can see here there's a lot less fusobacterium in the xenografts that have been from animals that have been treated with metronidazole. Not only that, but there's a lower level of BRDU incorporation, suggesting lower proliferation. And here we see an untreated in red versus in blue. The left panel is a control. It's a tumor cell line, HT29. The right panel, a patient-derived xenograft, COCO36, and we can see a significant slowing for this one xenograft in multiple mice when upon suggesting impact on fusobacterium treatment on tumor growth. So what do we confuse at this point? Sentence that was in our original paper. Both authors and reviewers wanted us to take, so we listened to the reviewers, but we fusobacterium cause cancer. Is it bystander or it's cancer? Our view at present is that fusobacterium could be liable for causing colorectal cancer in a civil suit, which requires a preponderance of the evidence. However, we believe that fusobacterium would currently be found not guilty of causing colon cancer in a criminal case, which requires evidence beyond a reasonable doubt. So thinking about mechanism, if any, does fusobacterium promote colorectal cancer? And on the next slide here, you see some data on the association of fusobacterium with overexpression of interleukins and other inflammation-related genes from Alex Cost's 2013 paper. Yellow are the high genes with fusobacterium. So is the relative expression by sample. High is yellow, low is purple, neutral is black. These are the most associated fusobacterium abundance. 1 beta, interleukin 4, interleukin 8, interleukin 6, CXCL5, other chemokine receptors, as well as PS2, OX2, the cyclooxygenase gene that's inhibited by non-steroidal anti-inflammatories, as well as 74 with L1. So on the next slide, just some questions in the future, bacterium and inflammation. Where does the expression of inflammatory genes, including interleukin genes, come from? Is it the immune or inflammatory cells? Is it the cancer itself? And does the inflammation promote the cancer or does it suppress it? So on the next slide, turning to analysis of some other cancer types. So this is work with Jax and Cheng Jen on mouse models of lung adenocarcinoma. And working with Spillman in my lab and other collaborators, Cheng showed that if you look at the number of tumor lesions, when you treat mouse under complete germ-free, specified pathogen-free condition, you can see a significant number of lesions as 8 weeks and 15. You can see here that when you treat with an antibiotic cocktail, compared to the left to the right, including metronidazole antibiotics, you decrease tumor burden. That when you have cancer-prone mice, it's a KRAS P53 inducible mutant, germline inducible mutant / knockout. You can see here in the significant increase in gamma delta T cells in the specified pathogen-free or microbe-containing conditions compared to the germ-free conditions, and also the and finally, it was a really interesting experiment. Cheng did treatment with anti-interleukin antibody associated with the gamma delta T cells and could show decrease in tumor burden and as well as in interleukin-1 beta messenger RNAs, suggesting that potentially one could regulate tumor growth here via disrupting an inflammatory process. So I think really interesting results. We do not see an association with the microbiome in the tissue of human lung adenocarcinomas, but this does to date. But this does not mean there might not be some kind of preventive role. I'd also like to turn to esophageal cell carcinoma, a work of Jason Nomberg in collaboration with Catherine Van Loon and Eric Cullison at UCSF, Susan Bowman at the Fred Hutch, and Elia Mumbaga in Dar es Salaam. Esophageal carcinoma is the most common cancer worldwide. Many of the cases are in developing countries, and you can see here higher incidence in the eastern corridor of Africa compared to other regions of Africa as well as other regions of the world. Jason and Susan did analysis of esophageal cancer, whole genome sequencing and RNA sequencing, and you can see here a high abundance both of Fusobacterium shown in blue and Prevotella shown in red. The data are concordant, as shown here by the Jacquard index, overall between RNA sequencing and whole genome sequencing data. And on this next slide, you can see another left C plot, enrichment on the top position here of Prevotella species, and second of Fusobacterium species in the Tanzanian esophageal squamous cell carcinoma compared to Cancer Genome Atlas North American esophageal squamous cell carcinoma, a work that's still in progress. And interestingly, in saliva specimens from the same tumors, you can see that if you pair by samples, you have an increased similarity between the tumor and the oral microbial the tumor and the oral microbiome in paired by samples versus random pairs, suggesting that there may be some influence of the oral microbiome on the tumor. Finally, I'd like to talk about a consortium that Wendy Garrett and I are leading, which is called the Opportunity to Investigate the Microbiome's Impact on Science and Treatment in Colorectal Cancer. It's one of the Cancer Research UK Grand Challenges. Our goals are these questions: microbiota promote bowel cancer risk? How does the bowel cancer host interact with the microbiota in human cancers and model systems? And what is the impact of the microbiota on the response to bowel cancer treatment? As I say, our collaborative team has a lot of colleagues, including Hans Clavers, Emma Allen Vercoe, Joseph Tabernero, Cindy Steers, at Dana Farber, including Marios Janakis and Kibbe Ng, Rob Holt, Curtis Huttenhauer, Shuji Ogino, Laura Porter, who leads our patient advocate team, Fiona Powry, and Phil Quirk. We've got a wonderful patient advocate team who've really been inspirational. Many of them most of them colorectal cancer survivors. I just want to talk about some work on the model systems from Hans Clavers' lab, using organoids to study the effect of the microbiome on colorectal cancer. And this is work of Cayetano Plezeguelos and Jens Pushoff and Axel Rosendahl-Huber, just recently published in Nature, led by Hans's group. So they've been looking at polyketide synthase positive E. coli, which is clinically associated with colorectal cancer onset. The polyketide synthase produces colobactin, shown here, which can make a DNA addict. They inject bacteria into organoids, and then when they both PKS positive and also E. coli with the PKS operon, but deleted for the CLBQ gene essential for synthesis of colobactin. And you can see that there's similar numbers of colony-forming mutants, but when they do gamma H2X and gamma H2AX staining, that's induced by the PKS positive, but not by the mutant bacteria. They looked at mutational signatures as a way to identify the ability of PKS positive E. coli to cause mutations. This is work pioneered by Ludmil Alexandrov and Mike Stratton's lab, and Mike Lawrence, now at MGH, working in Getty Getz's lab. And when they study the mutational signature, basically, they do whole genome sequencing of the colorectal, of the colonic cells in the organoids from both PKS positive or CLBQ mutant bacteria induced, and they compare the mutational signatures, and they find a clear mutational signature, mostly T to C substitutions at specific locations by this comparison. These mutational signatures are enriched in colorectal cancers from the Hartwig Medical Foundation cohort, published in Priestley et al. In Nature last year. And you can see almost all of the cases enriched are in the colorectal cancer, one in head and neck and one in urinary tract cancer, both with an indel signature and with a single base substitution signature. So just conclusions on this part: PKS positive E. coli induce mutational signatures in healthy intestinal organoids. These same mutations are seen in subsets of colorectal cancer patients. So one wonders if PKS positive E. coli can be used in the detection or prevention of individuals at CRC. And I think it's sure that any sorts of probiotics ideally would not contain PKS positive bacteria. Another feature of the work is a national network to study the microbiome on colorectal cancer treatment, led by Kimi Ng and Mark Janas at Dana Farber, Josep Tallero at Barcelona, to study early patients, patients with immunotherapy, patients with chemotherapy, and gathering tissue, blood, stool, and analyzing clinical data on treatment outcomes, diet, and lifestyle. And Kimi and Marios and Joseph have now really initiated a harmonized cohort, and they've begun enrollment at all of these sites, obviously limited today, like all clinical research, by the difficulty of the coronavirus epidemic. So just in some summary on Fusobacterium, I think we have more questions than answers. We see a strong association between Fusobacterium and colorectal, hints of the association response to antibiotics, a new association with esophageal carcinoma, intriguing from the JAX laboratory on pathogens in lung adenoma, so intriguing recently published data from Leiter's laboratory as part of our optimistic consortium that PKS may predispose to colorectal cancer. And again, our big question in the relationship between bacteria and inflammation. So I'd like to close now and take questions with really a special thanks to a number of people: Susan Bullman, now at Touch, Jason Namberg, my lab, Alex Kostich, now at Harvard Med, Ami Bhatt, now at Stanford, and a number of collaborators, including Wendy Garrett and Curtis Huttenhauer, long-term peers, Joseph Tabernero, Andy Hans Clavers, and our entire Cancer Research Grand Challenge team. And I just want to thank you very much for your attention. And I'm now very happy to take. Yeah.
Matthew, I can run a couple that I noticed during the talk. And then if you see others, that would be great as well. I guess one question comes up: has anyone looked in the setting of Lynch syndrome, familial adenomatous polyposis, for the presence of these bacteria, or if there's any association?
Sorry, can you repeat the question again? I just wanted to make sure I answered correctly.
Any association or findings of Fusobacterium in patients with Lynch syndrome?
So our colleague Cindy Sears has looked at early lesions in Lynch syndrome, and my understanding from her study, I think the first author is Deje, D-E-J-E-A, in Science, maybe about 2 years ago, is that when she looks at the early lesions, or actually she looked at precancerous lesions and saw an enrichment of Bacteroides fragilis, precancerous lesions, but not an enrichment of Fusobacterium, to my recollection.
Yeah, so I guess one question that comes up about the absence of bacteria in the cell lines is the obvious point that they're cultured long-term in the presence of antibiotics. Is anything known about if you propagate cells for a long period of time from colon cancers in the absence of antibiotics, whether you can generate lines that maintain some dependence on the bacteria?
I think that's a great question. It's definitely the antibiotics are preventing their growth. In short-term cultures, it's a technical issue, right? Of how do you prevent bacterial overgrowth in the short term? Maybe it would be worthwhile to take the xenografts where we still see the presence of the Fusobacteria. If we could take the xenografts and grow those, that might be a better way to do it, because then we don't have the high ground of bacteria from the colon. The cell lines is at least in, what I don't is ability of propagating lines derived from bacterial-free compared to bacterially present tumors. The fact that those cell lines that can grow without bacteria obviously shows that they don't require the presence of bacterial for their continued growth.
There's a question from Bob Weinberg. What specific laboratory animal would best be used to interrogate the role of Fusobacterium and Prevotella in colon carcinomas?
Bob, you'd be listening, first of all. And so I think I think that's a great question about the right laboratory animal. We've obviously been relying on mice and mouse models. I think that I actually think mice would still be the best, even though they don't. Many of the mouse models of colorectal cancer are not colorectal cancer prone. A lot of them develop rather than colon cancer. That's probably underlying Bob's question. However, I think that the availability of so many genetically engineered mouse strains still makes mice a great model studying. That still pick the best model.
So I'll sort of merge a couple questions together from Gordon Freeman, Stu Orkin, and Nathaniel Gray and others. Are the bacteria intracellular or extracellular? Is it really understood how they either escape from the immune system or might modulate immune reactions when they're in the blood and leading to metastasis?
Absolutely. So first, I'm going to question of intracellular. Fusobacteria are known to have the capability to invade cells intracellularly. We've shown convincingly that they'll invade colorectal cells in culture. Our histological analysis to date is consistent with them being intracellular, and our sequencing analysis, which I didn't show, we see Fusobacteria associated with individual colorectal cells in single cell sequencing experiments. Those two experiments are consistent with the bacteria being intracellular. We do not have the analytic resolution yet to establish that. I think using high resolution microscopy methods, we should be able to assess it. I call work in progress now.
Okay,
there was another piece of that was in Gordon's question, which I saw written down, which is, do the BECA produce anything that modulate the immune system and influence immune cells? I don't know. I don't know the answer to that, but we definitely see changes in the immune cell population the tumors, for example, with Fusobacterium, the colon tumors associated with Fusobacterium have more myeloid-derived suppressor cells in them. That's in our 2013 paper with Wendy's lab. Again, there are some reports on the Fusobacteria directly influencing this, and I know some groups are working on it. So I'm guessing the answer would be yes, but I don't know the answer. I just want to mention another question that's just adjacent to Gordon's in the list. It's from Berku Karadal. The question there, do you think the bacteria may be causing tolerance to tumors? I just want to say something I've wondered about a lot. I think it's a really interesting question, right? You know, how could the bacteria provide an advantage to the tumors? And I think that's a great question in that regard.
So Matthew, I guess there's an opportunity based on large numbers of data sets that are being generated in rare tumors. You didn't have time to discuss BPDCN, but that's a great example to do this kind of discovery. Do you have any guidance for how people who are planning to do sequencing of rare tumors might be able to interrogate their data sets or generate the data in a way that would be most useful for being able to identify associations with bacteria or other organisms?
Yeah, that's a great question, David. So yeah, we had a really nice collaboration. So Jim DiCaprio's lab and mine with Jason Umber, with Andy Lane, who studies blastic plasmacytoid dendritic cell neoplasm, and that's a cancer that seems like it would be it's associated with an interferon signature. There are a lot of pieces of evidence that made us think, this could be a viral cancer, and so we asked. We find either bacteria or viruses in association, and the answer was no. So in terms of doing these experiments, I think the first thing is making the sample clean and then as contamination-free as possible. So just collected and stored in a way that doesn't promote contamination or and I think that's incredibly important. That's the first thing that I think is key. I think the second is, consider, in collecting, we've actually made a lot of cancer types and different human diseases, sarcoidosis and others. We thought it was very plausible to try to identify associated bacteria or viruses without finding them. I think looking for epidemiological evidence, whether it's evidence of transmission, obviously would be incredibly important. And in the absence of epidemiological evidence, sort of physiological evidence, I think is important. I think it's just really important to do really careful analysis and comparison. There are huge number of false positives in the literature where I think people have, overemphasized, results that turned out later to be statistically insignificant or not important. There were a number of reports on SV40 and SV40 and mesothelioma, for example, that were using kind of error-prone methods. And so, it's just being careful and rigorous and checking it.
And can you comment? Are you aware of ongoing clinical trials using antibiotics, either in the context to prevent progression or, based on the data from Todd Golub and others, that the bacteria could modulate responses to chemotherapy or other agents?
Oh, I don't know the answer to that question. It's something, in our consortium, we've been exploring doing microbial replacement therapy. There are anecdotal reports of people using antibiotics. I don't know of any where they've also reported a benefit for using antibiotics in colorectal cancer therapy. So I don't think it would be unreasonable for there to be a clinical trial. I'm just not aware of one yet.
All right. Well, I really want to thank you for just an outstanding talk at the absolute forefront of science. We again had most likely well over 1,500 participants. As I mentioned, we will take a break tomorrow and then reconvene at noon Eastern Standard Time on Thursday for a lecture by Stu Orkin from Dana Farber and Boston Children's Hospital. Again, the lecture will be posted online at the same site for the Connect Science lectureship, and you should feel free to download it. That'll most likely be online later today, where you can also see the list of speakers who will be speaking over the next few weeks. If there are any further comments, please feel free to leave them through the site. Dr. Meyerson will go through the remaining questions and maybe in touch one-on-one with people to try to address any outstanding questions. So thank you all again for joining, and please spread the word. We'd like to have as many participants going forward as possible.
And I just wanted to jump in quickly for anyone that has not been to the website and maybe just got the link from an email. The website is dfhcc.harvard.edu/connectscience. That's where we'll post all of the upcoming talks.
Well, thank you, David. Thank you, Nellie. Thank you, Richard. And thank you for everyone who's been listening.
Alrighty, thank you. I'm going to hit end on the meeting now.